Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is the last lecture of this course, which is lecture number 63. So, this is molecular farming. So, where I will discuss the production of human somatotropin hormone in plants, particularly in tobacco. So, the concept covered is production of human somatotropin in transplastic plants. So, as you understand from this slide that so the approach is basically the gene to be targeted into the plastid. So, somatotropin, why somatotropin? So, there are few questions. Somatotropin, human somatotropin. So, the abbreviation is HST. So, somatotropin is basically produced in the pituitary gland, and what happens that this is basically the final form of hormone. So, the somatotropin which is produced in the pituitary gland will undergo post translational processing which includes this disulfide bond formation and eventually the processed HST is produced. PROC is processed, which is biologically active. Now, point is this, that when this one theoretical aspects let me tell. In pituitary gland, the removal of signal peptide from somatotropin, which leaves phenylalanine as the N terminal amino acid. So, in pituitary gland, removal of signal peptide from somatotropin, it leaves phenylalanine as the first N terminal amino acid. Now, if but in the plant system, what happens in the normal plant, it is basically the methionine, wherever in the plant translation process the first amino acid is methionine. That means, the normal translation in plastid basically that initiates at methionine as first N terminal amino acid. So, this to be taken care of if it should be of animal type. So, the methionine should not be the first N terminus amino acid. Therefore, if the gene for human somatotropin is available, say HST, So, if the HST gene is there, so this to be modified in such a way that it may be to be added with another protein which is ubiquitin. So, ubiquitin HST fusion protein to be made, where this will make the first N-terminal amino acid no methionine. So, rather I put here okay. But if only HST is used in the plant system, so in normal course it will make methionine as the first N-terminal amino acid. So, therefore, the modification has been made in such a way that a ubiquitin gene is fused, so that it encodes other amino acids apart from methionine. So, that is important. So, that modification they have made, and other important thing what they have done, they have used two different type of promoters. One is a strong promoter and another is a very strong promoter. The strong promoter what they have used, that is PSBA, and the other one is called P or RN / G10L. So, PRRN G10L is basically a much stronger promoter. So, basically it is ribosomal RNA operon promoter, and G10 is the leader sequence. If that promoter is there, so that is expected to provide much more stronger expression than the traditional PPSBA. Okay. So, this is only the things I need to mention, and then we will go to the construct. So, there are three constructs showing here. The first one is basically the without any modification, that means normal human somatotropin under the control of this PPSBA promoter and this terminator RPS16. Okay. And the second one is basically the fusion, but under the normal promoter, strong promoter PPSBA. But the third one is basically it is a much more stronger promoter, which is PRRN, GTL and the TPS. So, there are basically there will be three type of transplastomic plants. One is unmodified HST, another one is modified HST with a moderate promoter, and the other one is the modified HST with a strong promoter. And then this to be checked what is going to happen. So, accordingly this is the vector where this is the multiple cloning site here. So, gene was inserted here, as you see it is here, and here basically they are showing the different fragments which was required for southern analysis, but we are not going to discuss that. So, simply showing here that a ubiquitin HST fusion protein is basically now in the vector, and in addition you have the AADA, because AADA is required for the selection. Okay. So, the next one is this, that okay, they successfully produce the transplastomic plants. They have done southern analysis, but I am not showing that, simply I am showing the accumulation of HST protein. So, the purified HST protein was used for making the antibodies. So, they have used, this is a western blot and they are showing the accumulation of HST protein here. So, there are different lines. So, first one this is basically the A and this is basically the B. So, A is a nuclear transgene NT4747 and the plastid HST in this is 4848. So, these things. So, these are basically from the plastid, and so what you see, the level of expression in the plastid is much stronger when you compare with this one. Okay. And the B is basically where they have used the stronger promoter PRRN G10L. So, that basically showing more stronger HST. So, this is basically they have standardized the process and how they could get the more intense signal process. Okay. So, that means through this Western blotting they have confirmed indeed the HST protein of correct size is produced. Okay. So, because the size will be different when ubiquitin is attached and when ubiquitin is not attached. So, that has been shown here through 2 bands. Okay. Next is that let us consider the transplastromic plants were successfully produced and they have confirmed the presence of this HST and HST fusion as the case may be, because there are three different sets. One is normal HST, one is HST fusion with a moderate promoter, another is HST fusion with a strong promoter. So, in all the cases the transplastromic plant produce the HST as appropriate, and then what is important that once the HST produced, it is important to see that whether they are processed or not, because as I have said that the somatotropin requires post translation processing, which includes disulfide bond formation. So, in order to check that, what they have done is that they have they run the HPLC of the protein, they have and where what they have seen that when the protein they treated with DTT plus or DTT minus. So, when you see that when it is treated with the DTT plus, so this is with the DTT plus, and when is the DTT minus, that is a difference. So, this difference indicates this is DTT minus. So, this difference basically indicates that indeed the reducing agent diethritol makes modification in the disulfide bond formation. If that disulfide bond formation is not there, DTT treatment will not show any changes. At its showing changes, that indicates that the disulfide bond formation occurs. So, that means the transplastromic plants, the HST is basically processed within the plastid. Okay. And next what they have done is basically they confirm this protein by this mass spectrometry, and next what they have done that they another important thing they have done is basically they check that if the plastid expressed HST is biologically active, that is their question. So, that means how to test that? When you have the purified protein HST. So, that HST they have used it in a cell line called NB2 cell line. So, the NB2 cell line was used. So, basically when this HST or UBHST protein was added. So, this cell line shows proliferation. So, that means that rapid proliferation, because somatotropin basically stimulates cell division. So, rapid proliferation of NB2 cell line was noticed when the UBHST or HST is used. So, obviously UBHST showed a better response than the normal one. So, this indicates that the transplastomic plant produced HST is indeed biologically active. Otherwise, if it is not biologically active, it will not show proliferation. Okay. So, that part is fine. So, the next part is that how much is the protein. So, that just to give you an estimate that when the gene was localized in nucleus, and this is the plasmid, and so the gene localized in the nucleus, and you put a signal peptide sequence, so that the protein moves into the chloroplast. So, there the expression of this protein in terms of percentage of total soluble protein is only 0.025%. Okay. Okay. Where this protein, nuclear transform protein, was targeted to endoplasmic reticulum, it is again much less. But when the normal one, that is the unmodified HST, was targeted to chloroplast, was unmodified, sorry, unmodified HST was gene was transformed into gene was used to make transplastomic plants. So, the transplastomic plants in the chloroplast, when they have checked, the content was 0.2%. Okay. And when HST fusion was used using a moderate promoter, this level raised up to 1%, and when strongest promoter was used, this level goes to 7%. You see from 0.2 to 7% how much fold it has increased. I think more than 35 fold or so, something like that. Okay. So, next point is that one important properties, as I have mentioned, that maternal inheritance. That means, the transplastomic plant's pollen should not contain the gene, HST gene. So, that is the advantage, that when the pollen are escaped, the HST gene will not escape along with that. So, in order to check that, they have done reciprocal crossing. What they have done? They have taken the seeds of wild type as well as the seeds from this transplastomic plants. Like they have chosen 4838, maybe that is the best line. So, what they have done is they have made crossing. First of all, this is the female sign and this is the male sign. So, they have made crossing between this, and you see here, when wild type female is crossed with transplastomic male, then what will happen? The seeds, when they started germinate, it germinates. But why it is bleached? Because all this contains, okay, I forgot to tell, all this contains pectinomycin in the medium. So, this medium contains spectinomycin or streptomycin. Okay. So, now you have the seeds, you grow it in the MS medium containing spectinomycin. So, what will happen in this one, what I am focusing on, this particular one. So, it may start germinate, then eventually it will bleach. Why? Because there is no AADA gene is there. So, the spectinomycin will be absorbed by the plant and immediately it will bleach the tissue. Okay. Because although you have the NT4838, but that is a male one which was crossed. So, the gene AADA is not there in this. So, it is basically maternally inherited. The pollen of 4838 was used in crossing of the wild type female. As a result of that, this recombinant is produced. So, where there is no gene. Similarly, in this one, this is both the wild type. So, there is no question of gene. So, eventually it will die. Now, let us see the case here. Here wild type is male. However, the female contains the transplastomic. So, that means the wild type pollen was used and that will basically fertilize the NT483, which is transplastomic. As a result of that, recombinant is produced where the AADA gene retains in the female side. So, it retained. So, when the crossing was made, the recombinant, it retained the AADA gene. As a result of that, it survived, it survived in the medium containing spectrinomycin. Same is true for this one. Here both one is male and one is female. So, both of them contain the transplastomic, but the more point is that female have the transplastomic. So, therefore, this one is also survived. So, this is called biological containment. So, these indicate that indeed these are transplastomic plants, and it is safe to grow the plants in the field, so that it will not cross pollinate other plants in the nearby field and lead to the creation of superweed. So, that is why we call chloroplast transgenes are maternally inherited. So, that property has been also tested. So, therefore, to end this one, so what I said that human somatotropin gene they have modified in such a way, so that it when the first translation amino acid upon translation, the first amino acid will not be methionine, but anything then apart from methionine will be produced, and they made this by fusing ubiquitin before the HST, and if HST is not there, it will make the methionine. So, because we want to make more of the animal type, because somatotropin not the plant type. So, therefore, this fusion was required and it worked well. So, the gene was successfully inserted, they have checked the size, they later checked the expression, and they also checked the HST fusion protein formation by Western blotting, and then the protein was tested for its post translational processing properties or not by DTT treatment. Then protein was also tested for its cell proliferation capacity. So, that also successfully passed, and finally, they have tested the biological containment, whether these are indeed the transplastomic proteins or not. So, that is that the plants are indeed transplastomic and the protein is produced in this. And we have also shown the level of expression is much higher than that of the nuclear transformation, and why that I have explained in the previous class. So, this is basically a very successful example of high yield production of human therapeutic protein in tobacco, and this paper published in Nature Biotechnology. I will just give you the reference, although this is an old paper, but this is the best paper to tell the positive results of transplastomics. So, this was published in the year of 2000, volume 18 in the March issue, page number 333 to 338. So, if you are interested, you can get this paper, simply this is available in the ResearchGate. So, if you and the first author is Staub, Staub, Staub et al. And this was basically this work was done at Monsanto company and Agracetus company. This is not from academic institute, but a private laboratory. So, Monsanto is the giant in agricultural biotechnology. So, they ventured into the field and they produce this work. So, and that this is a very successful case of transplastomic plants, and particularly for the teaching in the class, I always prefer to discuss this paper rather than other papers. So, that students can really understand the beauty of this transformation. So, with this I end this class and I end this course. Hope you have enjoyed this course, although a bit heavy, but I try to cover a lot of things, but of course, there are few things which remained left out, but maybe in future when I will modify this course I will add it up. So, hope you will be able to make to submit the assignments, and I expect that a sizable number of the students will write the exam and they will collect the certificate, which may be useful if they are interested to go for higher studies. So, such certificate will be useful, and at least because this is a full course, and you will be able to answer questions if asked during your PhD or integrated PhD interviews, or wherever may be postdoctoral anything cases. So, knowledge will always be wealth for you. With this, I end this class. Thank you very much for your patient learning going through the process. Thank you.